Can you tell me which one of these animals has stripes?
This one has stripes.
The question in this demonstration is simple to answer, unless you have a disorder called Pick's disease.
It's a cat.
Pick's disease is a form of dementia in which people gradually lose knowledge.
This is a horse.
First, people lose the ability to make subtle discriminations.
This is a horse.
Such as how one animal differs from another.
And which one has horns?
Horns. This one has horns.
But they retain concepts with more general differences.
Just tell me, which one of these things is not an animal?
This is not an animal.
Why is Pick's disease of interest to psychologists? Dr. James McClelland is co-director of the Center for the Neural Basis of Cognition, a joint project of Carnegie Mellon University and the University of Pittsburgh. And he says Pick's disease serves as a model for understanding key points about the brain and cognition. First, knowledge is distributed throughout the brain.
There's one part of the brain where, when we think about the color of something, that part will be activated. Another part, when we think about the shape, that area will be active. Another part where if you think about the sounds that it makes, that area will be active.
Second, thinking is related to brain neurons.
Our cognitive activity arises from the neural networks in the brain.
Third, the networks and the knowledge in them change gradually.
Whenever you lose an individual neuron, you're not losing an idea. You're just losing a tiny bit of the resolution or the crispness of that idea.
And fourth, by simulating neural activity with computer models, we can learn more about the brain, cognition, and memory.
Okay, Mr. Hunter, this is going to go over your head.
As shown in this demonstration, a number of high-tech procedures are used to understand cognitive activity in the brain in general, as well as in Pick's disease.
You're going to feel the table go up.
This procedure is a magnetic resonance image, or MRI.
Okay, the most important thing is you have to keep your head extremely still. Once you get in there, we're going to take a few structural images.
The patient slides into a 4-ton magnet.
Are you okay, Mr. Hunter?
Yes, I am.
The resulting structural images show the extent and location of brain damage.
You can see in orange here how the damage has affected the temporal lobes on both the right and the left side of the brain very extensively.
As damage to the temporal lobes gets progressively worse, there's a corresponding loss in the patient's knowledge.
This one's a cat.
MRIs can also show the brain in action.
Okay, Mr. Hunter, we're going to go ahead and try this word task.
When the patient sees a word on a small mirror in the scanner, he thinks of a word associated with it, and the MRI shows his brain function.
The red-colored area represents activation of the brain that's working while performing this task.
Brain areas other than the damaged temporal lobes may look normal, but not function correctly. But McClelland is interested in getting a finer-grained look at Pick's disease and cognition in general. It's a look that can't be seen on a brain scan, a look that takes him to the level of neurons.
These are the individual nerve cells, but each of them has a very extensive and complex branching structure that allows it to receive inputs from many, many other nerve cells. And when you think about your ability to have a thought or to produce an action or to say the name of a word, we think in terms of the idea that there's simultaneous activity over a large number of these neurons.
McClelland can't study real brain neurons while people are alive, so he's designed a computer model.
So this is a picture of one of our simulated neural networks. It consists of a number of artificial neurons, and each of them has connections that they receive from other brain areas, and also they send signals out to other brain areas, and they're also fully interconnected with each other.
The network is taught to think just as humans are, and it can be damaged as if it had Pick's disease.
I can damage nerve cells in the simulation in the same way that we think they're damaged in the brain, and we can.
As individual neurons die, one after another, the network's knowledge gradually deteriorates.
It loses the ability to answer the questions that differentiate very specifically, let's say, between the lion and the tiger, before it loses the ability to say, oh yeah, these things are all animals and those, are not.
So the network mirrors the gradual decline in the patient.
It's an animal, but I don't know what kind of animal.
Will the mystery of Pick's disease be solved by artificial neural networks? That's hard to say, but they're helping us to understand Pick's disease as well as overall cognitive development.